Hey guys, this is Mr. Sodal, and we're about to begin the Chapter 2, Section 3, Chemistry of Life, Macromolecules PowerPoint. You can see there's some key concepts here on the bottom of the page to keep us focused throughout this PowerPoint lecture, and make sure that you're referencing them as we are continuing through this video. The objectives of this PowerPoint are first to provide two reasons to explain why carbon is so versatile, to explain the difference between dehydration synthesis and hydrolysis and the role that water plays in each reaction, to identify the macromolecule grouping of a picture of an organic structure. This is something you'll need to be able to do on the test quizzes in Keystone, so make sure that you are paying attention on how to identify these macromolecules. Describe the following for each macromolecule group: the key elements it's made of, overall function, monomer examples, polymer examples, type of bond formed during polymerization, uniquely identifying characteristics, and distinguish it from other macromolecule groups. First, why is carbon so versatile? Carbon is versatile because it can form 4 single bonds or 2 double bonds with a variety of different atoms, even other carbon atoms. Carbon is a very versatile atom, necessary for life. Carbon can bond to itself, allowing the formation of long chains and ring structures. And compounds that contain carbon are called organic compounds. We are made of organic compounds, and all life on Earth relies on organic carbon-based compounds. Monomer versus polymer. These are two terms that are very important to be able to distinguish. Monomer is the term for a small molecule that can join together to form a larger molecule, the polymer. It's like the individual links in a chain. The prefix mono actually means one, and that's why it is one monomer, a smaller molecule. The polymer, prefix poly meaning many, is a large molecule made of repeating smaller molecules, or the monomers. The polymer in the chain example would be the entire chain. Okay, here we see that in this picture, and then in what's called a structural formula for a polymer. Okay, you see each individual ring is a monomer structure that are joined together by a bond to form the polymer. The next topic is dehydration synthesis versus hydrolysis. Dehydration synthesis is the removal of water to link two smaller subunits together to form a polymer, whereas hydrolysis is the addition of water to break apart large polymers into smaller subunits. This diagram shows dehydration synthesis. Here we see a monomer molecule and another monomer molecule. If we remove this oxygen atom and these two hydrogen atoms, which makes H2O, water, we can then form a bond between this carbon atom and this oxygen atom, linking those two molecules together, thus making a polymer. The link here at the bottom of the page, if you click on that, shows an animation that highlights the difference between these two processes. Hydrolysis is simply the opposite of dehydration synthesis, where these two hydrogen and one oxygen atom are added back in to a polymer to split it apart into a few monomers. So, macromolecules are very important living things. There are 4 categories of macromolecules that we will be discussing: carbohydrates, proteins, lipids, and nucleic acids. We'll start by discussing carbohydrates. This would be a good time to make sure you're following along on your handout, and this information would be going in the chart that is a part of your handout. Carbohydrates contain 3 atoms: carbon, hydrogen, and oxygen. And they are found in a ratio of 1 carbon to 2 hydrogen to 1 oxygen. So basically, however many carbon atoms there should be twice as many hydrogen atoms and the same amount of oxygen atoms in that molecule. This is a good way to identify carbohydrates by their structural formula pictures. The monomer of carbohydrates is called sugar. The technical term is monosaccharide. Some examples of that would be glucose and fructose. Polymer of carbohydrates is starch. technical name polysaccharides. Some examples are cellulose, which is plant starch, makes up plant cell walls, and glycogen, which is animal starch, which is found in all of us. The type of bond that holds carbohydrates together is called a glycosidic linkage, and the function of carbohydrates are as chemical energy sources. They're used for plant cell structure, I mentioned the cell wall, and they're also used for cell identification. Carbohydrates are found on the outside of cell membranes and are used for cells to recognize each other. This page shows some example carbohydrate structural formulas. Here's a monomer example. This is a very common sugar, glucose. See its formula C6H12O6. There's that 1 to 2 to 1 ratio. If you were to simplify these numbers, for every one carbon, there's twice as many hydrogens and the same amount as many oxygens. 6 to 12 to 6, 1 to 2 to 1. Notice we have carbon atoms forming a ring here, because carbon is so versatile. Carbon is making 4 bonds. One bond to this carbon, another bond to this carbon, one bond to an oxygen, and another to a hydrogen. Okay, but notice not every atom can make that many bonds. Hydrogen only makes 1 bond, oxygen can only make 2. Okay, here's a polymer example, amylose. And amylose is a starch found in a lot of our foods, okay, and our body can easily break it down. And we see it is made from joining the ring structures of glucose together. And if you look closely, this is pointing at that glycosidic linkage. The bond between the carbon and this oxygen here is considered a glycosidic linkage. So between each subunit, we have one of those bonds. The next type of macromolecule that we are going to study is proteins. Proteins are very important for living things. Proteins are made of 5 different atoms, and maybe a 6. Carbon, hydrogen, oxygen, nitrogen, and sulfur. CHO, CHON, is what they are typically consist of. Very few proteins have sulfur, but sometimes they do. They are made of a monomer called an amino acid. Amino acids are very essential for our survival. Okay, there are 20 different types of amino acids that are important to humans, and we get them from the proteins that we eat. Polymer, polypeptide, is the name for a large protein when it's joined the amino acids together. An example would be hemoglobin, which is the protein found in our blood. The bond type here is called a peptide bond that holds the amino acids together. And proteins have many functions. Just to name a few important ones, they're important for cell structures, muscle structure, our cartilage and things like that are made of protein. Enzymes, which help speed up our chemical reactions. We'll learn a lot about enzymes this year. Antibodies, our body's movement, and there's, like I said, many more functions for proteins. Straying from our chart a little bit, okay, each amino acid, we said there's 20 different ones. The reason that they are different, or where they differ, is in the R group. Okay, it is different for each amino acid. Okay, we can see similarities in each of these amino acid structures. We have this amine group with the nitrogen and 2 hydrogens, a central carbon atom, a central hydrogen, and a carboxyl group with a carbon, an oxygen, and an OH group, which is oxygen and hydrogen. But then this green section is different. So for the amino acid glutamic acid, we have this side chain, histidine, we have this one, and glycine is simply just another hydrogen atom. This structure down here would be your polypeptide. There's our central carbon, here are our groups, okay? Nitrogen, carbon, carbon, nitrogen, carbon, carbon. The bond between the carbon and the nitrogen is the peptide bond. Okay, and there's our polypeptide. And the peptide bond between the carbon and the nitrogen. The sequence of amino acids in the protein chain is determined by our DNA code. We're going to learn more about the significance of how DNA is used to produce proteins later in this year, but it's important to know that each protein is unique and created from our genes, and it is what really determines our traits and characteristics and what makes each human unique, and each organism really unique. Proteins also have a 3D shape, and the shape directly affects its function. We're going to talk a lot about this with enzymes, okay? And there are things called hydrogen bonds that hold the proteins together, okay? And if protein shape gets ruined or misshapen, then the protein will not be able to function properly. So we've learned about carbs, learned about proteins. Our next macromolecule is lipids. Lipids contain carbon, hydrogen, and oxygen. Very little oxygen, not the 1 to 2 to 1 ratio that we saw in carbohydrates. The monomer for lipids is actually no true monomer. They're typically made of these two components, glycerol and fatty acids, but not every lipid is composed of them, and lipids are similar in many ways, but they are not made of repeating subunits like carbohydrates or proteins or nucleic acids, as we will see in a moment. Polymer for lipids is no single name. There are several types, as you can see here in the examples. Fats are an example of a polymer, oils, waxes, cholesterol is a type of lipid, and phospholipids, which is an important lipid in our cell membranes. The type of bond that holds the lipid smaller subunits together is called an ester bond. And lipids function primarily as energy storage. That's the fat that we store. Okay, they actually create cell membranes, as I mentioned with the phospholipids. They're used for insulation, the blubber of whales, for instance. And lipids are also used to make certain hormones, okay, that can be used to control cell processes and for cell communication. Lipids are a non-polar molecule. We've learned about polarity in the water section, okay? Lipid does not have a positive end and a negative end. It's a even charge, a neutral charge throughout. And it's because they have very few oxygen atoms. And because lipids are nonpolar, it means they do not get along with water. They are what are called hydrophobic. Hydro standing for water, phobic standing for fear, meaning they have a fear of water. And therefore, they are insoluble, or they do not dissolve in water. Okay, as we can see in this picture here, we have water on the bottom, oil on top. Oil and water do not mix because oil is not polar and water is polar. This structural formula we see here is one for a molecule of cholesterol. Notice there are lots of ring structures here, okay? But when we look, I only see one oxygen atom. One oxygen atom. Carbon, all these ring structures have carbon around them, lots of hydrogens, but only one oxygen. Some structural formulas for lipids. This is a typical fat structural formula. We see 3 fatty acids. That's why sometimes fats are called triglycerides, because they have 3 of these fatty acids here. This fat would be what's called an unsaturated fat because it has this little kink here. You may have heard of those. We'll talk more about those in class. This is a structural formula for a phospholipid. The phospho comes from the element phosphorus, which is added to it. Notice it only has 2 fatty acid chains. We'll learn more about them when we learn about cell membranes and cell membrane structure. This would be the formula for a wax. Again, notice only 2 oxygen atoms. All the rest is carbon and hydrogen. And now we are ready to move on to our final macromolecule, which is nucleic acids. Nucleic acids contain the most atoms. They contain carbon, hydrogen, oxygen. Notice how these 3 were in every macromolecule. They contain nitrogen and phosphorus, sometimes referred to as CHONP. Okay, making a little acronym out of the element names might help you remember them. Okay, nucleic acids are composed of CHONP, whereas proteins are made of CHON, and carbs and lipids are made of CHO, CH&O. The monomer for nucleic acids is called a nucleotide. Okay, and there are 4 different nucleotides that are used to create the polymer of nucleic acid: adenine, thymine, cytosine, and guanine. Polymer is called a nucleic acid, and there are 2 main nucleic acids found in living things: DNA and RNA, which I'm sure you've heard of. The bond type that holds the nucleotides together is called a phosphodiester bond, and I'll point one of those out to you in a moment. Function of nucleic acids, DNA, is to store and transmit genetic information, and they also contain the instructions for protein synthesis, which is a very important part of our characteristics, our traits, and our genetics. Okay, without DNA, we would not be able to pass on traits to our offspring, and therefore they would not be able to be produced. This is a very general diagram of a nucleotide. Nucleotides are made of three smaller structures. Okay, a nitrogen base, which is a molecule containing the element nitrogen, a sugar compound, which has our CH and O, okay, and a phosphate group. Here's where the element phosphorus comes in. Changing the sugar and nitrogen base produces different nucleotide subunits. Every nucleotide will contain phosphorus, a phosphate group. If it's DNA, it'll contain one type of sugar, RNA, a different type of sugar. And even within the DNA and RNA, the nitrogen base can be changed, as we'll see in a moment. Nucleotides. Again, we can have two sugars used. Deoxyribose is the sugar found in DNA, and ribose is the sugar that is found in RNA. Notice they're very similar. The only difference lies right here. This hydrogen versus this OH group, oxygen hydrogen. Okay? Ribose, deoxy, one less oxygen than ribose, DNA. Okay? Has one less oxygen. There are 5 nitrogen bases: adenine, guanine, cytosine, which are found in every nucleic acid. Thymine is only found in DNA, whereas uracil is only found in RNA. We will learn a lot more about these, their pairing relationships, and how they affect our traits later in the school year. Again, there were 2 types of nucleic acids: our DNA, deoxyribonucleic acid. DNA is double-stranded. And notice here the pairing relationships. Cytosine pairs with guanine, thymine pairs with adenine. And they can repeat, and this pattern is what makes each of our DNA sequences unique, and therefore our traits and our genetics unique. RNA stands for ribonucleic acid. RNA is single-stranded. Looks like half of a DNA molecule. Notice it has A, U, G, and C, not thymine, no T. Structural formula for nucleic acids. Here we see a molecule of DNA. Here's the familiar double helix shape. If we zoom in to the structural formula, we see there are several ring structures. Okay, this would be our nitrogen base right here. Okay, bonded to the deoxyribose sugar and then the phosphate group. These bonds between the sugar and phosphate right here, that would be our phosphodiester bond. Okay. And these are hydrogen bonds that hold the nitrogen bases together and therefore the strands of the DNA together. Here's a zoomed in picture of a nucleotide, the monomer. Here's our nitrogenous base, adenine. Notice all the nitrogen atoms in there, hydrogen, and then the omitted carbon atoms. Sugar, phosphate group. Oops. This would be our RNA molecule. Okay. See, as it's single-stranded, there's nothing connecting these nitrogen bases, which are on this side of the molecule. Here are ribose sugar molecules and our phosphate groups. Okay, identifying the nitrogen, the carbon, the oxygen, the hydrogen, and then the phosphate will allow you to identify these structures as nucleotides. Nucleic acids. And last but not least, we have our macromolecule structural summary chart. You can use this to compare with your chart that you had filled in throughout this presentation. Okay, our macromolecules are listed here: the general monomers, the type of bond, the general polymers. Okay, and the functions were all discussed throughout this PowerPoint. Okay, remember, you can pause and rewind and watch this over again if you've missed something. And then, as always, come to class with your questions. Hopefully, as you were going through this, you were making some notes where you did not quite understand something, so that we can go over it in class and I can help to, help you understand this information. Okay? Thank you, and look forward to talking about this in class with you all.